Welcome to Discover ASAP, the video interview series where I connect with members of the ASAP network to talk more about the different research outputs that they have been utilizing and are producing. And so for today's video, I have with me Koen Theunis, who's a technical expert at the Laboratory of Computational Biology at KU Leuven and part of Team Voet. Good morning. Hello, Koen.
Good morning. Evening for me, but hello.
Yes, we're on two different sides of the world here.
Yep.
So before we talk more about the specific research output, which just for everyone's awareness, we're going to be talking about something called HyDrop. I was hoping we could just take a step back and talk more about Team Voet's ASAP project.
Okay. The project starts actually from the idea that the majority of disease-associated variants are located in the non-coding genome. And their functional effects remain challenging to decipher, actually. And to study the effects of non-coding genetic variation on gene regulation, we therefore generated a single nuclei, actually, multi-omic atlas of the human cingulate cortex and substantia nigra, and other other labs in Team Voet, for example, focus on the gut to create a multi-omic atlas. And additionally, we also have matched long-read whole genome sequencing data to get the genetic variation of each of the donors. That's basically the general idea where we start from. And then the single-cell multi-omic atlas is built from single-cell. It's using both single-cell ATAC-seq data and single-cell RNA-seq data. Most of it is from a multi-omic assay of 10x genomics, but we also use standalone assays to get more cell numbers, nuclei numbers in the atlas. Yep.
Thanks for providing that description. So today what we're going to really focus on is something called HyDrop that was developed by Florian de Roop, who was a previous member of Team Voet, and thank you for stepping in and discussing it with us. So Can you just tell us more about what HyDrop actually is and what the motivation was behind creating it?
Yeah. So basically HyDrop, it's like a droplet microfluidics technology, and you can use it for droplet, single-cell droplet technologies. So the most famous is a commercially available 10x genomics, for example. So in that case, it's very simple, but similar. But yeah, this one is open source, an open source platform. And the main motivation for us to start this in the past was you had also other technologies like DropSec. So I think that the lab before I was there also already was using a DropSec system. That's how they got familiar with Droplet Microfluidics. But what was lacking at that time was, for example, a single-cell ATAC-seq assay for Droplet Microfluidics. So that really was missing at that point. Now there's also the 10x kit. That's where you can get your single-cell ATAC-seq data. But when they started developing HyDrop, this was not there. And then the other thing was to reduce price and increase quality of droplet single-cell RNA-seq compared to the standard at that moment. Then we also wanted to keep everything open source. So we have protocols at .io, protocols for all the steps that are needed to get this running.
Yeah, amazing. So you've mentioned, that you guys have these protocols and it's open source. So if other labs were to want to try and use HyDrop, what actual elements would they need to enable them to actually perform it?
Yes. So one of the, let's say, uncommon devices or equipment that you need is like a pump system. So this is needed to generate droplets. Then the next part would be a microfluidic chip. And this you can either make yourself with the protocols we provide, or you can also buy these commercially these days. So you need the pumps to pump the oils and everything through the microfluidic system. And then another big investment or a big thing that you need to create are these barcoded oligos, to barcode all your droplets uniquely. So this could be like a big initial investment, but once you have the plates with the barcodes, you can make thousands of runs with those plates, because it uses such small amount of oligos. And then lastly, depending if you want to do single cell RNA-seq or single cell attack-seq or something else, you need normal components that you would otherwise need for, let's say, a plate-based method. Yeah.
So you described the microfluidic instrument, and I know I was reading your guys' protocols that you guys use the Droplet Genomics Onyx microfluidic instrument. So
Yep.
have you tried using other setups, and if so, were they successful?
Yeah, I know that initially, instead of using the Onyx, because the Onyx from Droplet Genomics is the instrument that we use now, they used also pumps from, I think, a company called Harvard, and the other, I wrote it somewhere down. I want to say it right correctly now. Well, in any case, there's acetone pumps. That's another pump system that has been used in the past, and they also work. But depending on which you use and which syringes you use and tubing, your flows will be more stable or less stable. And in the end, we've now been using for several years the Onyx system. And the additional advantage of that is that because you also can use or need a microscope to visualize your microfluidic system, and this is integrated in the Onyx system, which makes it all a bit more compact and ergonomical. Yeah.
And
But
you,
we had successful runs with the other systems.
okay. Oh, okay.
That's how it was. I initially started developing, and yeah.
Okay, great. That's good to know. You mentioned a few times, we've alluded to it as well, that you guys have, created these series of protocols to be able to actually perform HyDrop. And so if someone was trying to implement HyDrop in their labs, and they're looking at these protocols, are there any like really critical steps or components or troubleshooting tips that you just want to make sure that other people are aware of?
Yes, I think one important thing is that you familiarize yourself with the pump system. So often when new people start in the lab and use our protocols, we just run simple droplet generation to get familiar, because you can adjust the flow rates, and this is a bit of a thing you have to learn a bit, but goes actually quite fast, since we only have 3 pumps that we have to keep an eye on, and most of these are kept stable during the run. It's basically like one rate that you can adjust over the run a little bit. So that's just a little bit of practice. A very important thing, of course, is that if you make the clonal beads yourself with the barcodes, is that you do this very rigorously, because in the end, the most important thing that you need in this assay is like a very bead that is as pure as possible. Because if there are impurities, or if there is much change in the size of your beads, for example, this leads to flow instabilities. So this is something that you might need to optimize. But up until now, I think we've been very responsive to questions on protocols.io. Mainly Florian has been keeping track of this. And I know that in the past, we've also helped a few labs in testing HyDrop in their own facility. So yeah, we're always open. We're super happy if people also want to try this, and because it can only improve these kind of protocols. Yeah.
Yeah, that's amazing to hear. Thank you so much. And thanks for providing that insight on the size of the beads. It makes sense when you say it, but it's not something that I think I would have probably initially thought about.
Yeah.
So thank you so much for joining me today and for chatting through HyDrop. Really appreciate you being here.
Okay. Thank you very much.